Welcome to our review on growth and development. So first thing we need to know then are the structures found in a typical animal cell and a typical plant cell. So best thing you can do here is just practice drawing them out and labelling those parts. So your animal cell should always have 4 parts: the mitochondria, cell membrane, cytoplasm and nucleus, whereas your plant cell should actually have 7 parts. So the nucleus, the chloroplast, cell membrane, the vacuole, the mitochondria, the cytoplasm and the cell wall. You also need to know what each of those organelles actually does. So some of these we've already looked at. So the cell membrane controls the movement of substances in and out of the cell. Your nucleus is going to control the cell. The cytoplasm is where the chemical reactions take place, and the mitochondria are where aerobic respiration takes place. We've got obviously 3 new ones there for our plant cells, which is the cell wall, which is basically made of cellulose and is very strong to support the cell. We've got the vacuole, which is filled with the cell sap, and that is all involved in supporting the cell. And then finally the chloroplasts, and these ones are filled with a green pigment called chlorophyll, and they're all to do with trapping light energy so that photosynthesis can occur. Bacterial cells are slightly different. So you can see in the diagram down there that they've got these bits sticking out the back called a flagellum, but they're not always present, okay? They act like a tail basically. Then inside the cell itself, they've got a cell wall, and they've got 2 types of DNA, okay? They've got it that occurs as a plasmid, which are those small orangey circles, and they've got the bacterial or chromosomal DNA, which is basically one big loop of DNA, a circular strand if you like. They've also got the cytoplasm there. Key thing to remember then about our bacterial cells is that they're smaller than plant or animal cells. They have no nucleus, no mitochondria and no chloroplasts, and the DNA occurs as a single circular strand. So when we actually think about growth then, what we start off as is a fertilised egg cell, and that fertilised egg cell is also an undifferentiated stem cell. Now what that means then, stem cells are undifferentiated cells in their nature, so that means that they have no particular function at that time, but they could become any cell type. So what we see initially then, starting with that single fertilised egg cell, then we have mitosis occurring to make identical copies of the cell, and then we have this process of differentiation occurring, which is where they become specialised. So what we see is as we make more and more cells, some of those cells will become differentiated into one type and others into another, until we finally make up a little human. If we think about human growth then, what we end up with is a very particular shaped graph, as you can see on the left there. So we've actually got 4 stages of growth in humans. The first stage is our infancy, and that's where we've got very rapid growth, as you can see from that steep line on the graph. Then we enter childhood, where we have a slower rate of growth, but it's steady growth. When we hit adolescence then, so that's usually teenage years, that's where we get this rapid growth when we go through puberty. And then finally, as you get to adulthood, then your growth rate falls to 0, because as a mature adult, humans don't grow anymore. When we think about plants though, we see a different situation, because unlike humans, plants will grow throughout their entire lives. However, they will only grow at these sections called the meristems, and those meristems are found in 3 specific locations of our plant. Firstly, the root tips, secondly, the shoot tips, and thirdly, in the buds. When we think about meristems then, again they're undifferentiated, so that means they've got the potential to turn into any cell type. They've got small vacuoles and very thin walls. They don't have any chloroplasts, and they're capable of dividing to make new cells. If they had thick walls, they wouldn't be able to divide, okay? So basically this is what's going to allow it to become any type of cell that the plant needs. Where we've talked about stem cells then, we can actually use them for medical purposes. So potentially we might be able to use stem cells to treat things like Parkinson's disease. We may be able to use them to repair spinal cord injuries in those people that become paralysed and give them the ability to walk again. We could even potentially grow entire organs or tissues for transplant. And finally, we might be able to use it to treat type 1 diabetes so that people don't have to keep injecting insulin. As with all of these things, however, even though they're good, then there are downsides and reasons that people may object to it. First one is it might go against people's morals or ethics. And secondly, it might go against their religion.